producing. So thank you for inviting me. It's a pleasure to be here. I'll tell you briefly about some of our experiences in synthetic biology. Actually, about 15 years ago, as a computer scientist, I became fascinated with the notion that we might be able to program cells as easily as we program computers. And so I'll tell you about some of our experiences going from starting from parts, modules, and now starting to think about some applications where synthetic biology, we believe, makes a significant difference, allows you to do things that you wouldn't not be able to do otherwise. And so this notion of being able to sit in front of a computer was presented in the previous talk, and that's many of us in synthetic biology would like to be able to do so. So think about some kind of a high-level behavior, specify it hopefully formally, and then use some computational mechanisms to figure out a sequence that actually implements that particular behavior. Send it off to DNA synthesis. A couple of days, couple of weeks later, you get a tube contains a DNA. You put that into the cell. It does exactly what you want, right? And then you can move on, retire. You know, we're not quite there yet, as you can imagine. There are a few things that we need to, kind of big challenges that we need to overcome. And certainly we need to understand the iterative process of design and implementation. How is it similar to what we know? How is it similar to what we do in electronic design, design computers, various other kinds of machines, whether it's planes, cars, or so on? And what is also different about the world of biology, the substrate of biology, for example, directed evolution. How can we use directed evolution to optimize this whole process? And then if we're successful, applications for synthetic biology include production of various high-value compounds, bioenergy, environmental applications. And today, second half of the talk, I'll focus on some biomedical applications where we're starting to make some headway. Cancer and artificial tissue homeostasis for beta cells. So I'll give you some flavor of what we mean by system-level design for these particular applications. And so what I'll do is I'll start from some parts to modules, briefly talk about work that we're doing on a BioCompiler, and this is with Jake Beal and Doug Densmore, who's actually, you mentioned Clotho, so he's involved in this. And then I'll mention some examples of larger scale systems and some of the issues that we encounter as we go to the larger scale. And so there's been a plethora of parts and devices that have been made available over the last 10 years or so. And many of them are available in the registry of standard biological parts. Those, for example, include regulation, could be gene activity, could be RNAi. So I'll talk about, for example, the inverter. I mentioned that. This is a part that we use quite a bit. Transcriptional repression, RNAi, I'll show that here. For example, Wendell Lim in our lab is also working on protein-protein interactions. So that's a very important area, I believe, as well. Cell-cell communication is quite important for many of the things that we do. So this dates back to the work I actually did for my PhD in the 90s, engineering cells to talk to one another with AHL. And there's been other examples, reporters. There's been many, for example, this is from Cambridge, and this is actually from Cambridge IGEM team as well. There's a lot of fun things you can do with respect to getting the cells to tell you what's going on, as well as interfacing. So, for example, one thing that many IGEM teams like to do is kill the cells. So this is with a whole protein, detect various inputs from the outside, whether it be light or control motility. So what you're seeing here is that kind of our toolbox of parts that are available is expanding rapidly and certainly allowing us to think about many fun and perhaps useful things that we can do with synthetic biology. And now we have to start thinking, and I think this is, in fact, the thing that differentiates synthetic biology from previous efforts is the systematic approach to taking these parts and then being able to construct larger scale systems out of them. And so I'll just mention briefly a few of the modules that we've implemented, and these we did, this is several years ago, and we used some mechanism where we did design by hand, but oftentimes we would then create computational models and try to understand how is the design that we created by hand implementing the specifications that we in fact started with. So for example, here we have a transcriptional cascade of inverters where the output is the not of the input. And the question that we're asking in this particular example is, do these biological parts actually allow us to exhibit and implement digital behavior? Does that meaning is the output a better representation of the digital meaning than the input? And so what you're seeing here experimentally, input versus output, that this is the first stage, the blue, and the last stage is this yellow, is that the output actually becomes more digital, becomes more step-like as the cascade gets longer. So that means we can take noisy biological components and actually create robust digital systems out of them. But not only do we deal with digital systems, we also worry about and try to design things that have temporal aspect, that have amplitudes and all kinds of analog properties. And what I'm showing you here is a pulse generator, and here we have cell-cell communication where cells are engineered to send an HL. So you heard about HL in the previous talk, where now HL activates with a feed-forward motif, a pulse of GFP. And the first implementation actually did not work, so we had to go back to the computational models and figure out how to re-engineer the system, mutate the various elements in order to get pulses that actually look nice. And that's the ones that you're seeing over here, guided by the computational design. And another quick example is where we now are using cell-cell communication, a feed-forward motif that is longer, to actually engineer spatial patterns. And so here's cells sending messages to receiver cells, and those respond only when we have intermediate levels of this AHL. And you see we can create a variety of different designs. And so what we're doing now is taking these basic principles and implementing them with embryonic stem cells, and the idea is to be able to create tissues by design. So I won't mention any work on that later, but we are starting to make some headway in terms of controlling the cells, in terms of being able to differentiate embryonic stem cells on command and so on. And so, and again, I mentioned the work with Jake Beal and Doug Densmore. So what we're trying to do, and that's also, as you saw in the previous talk, is fully automate this process, where we have some kind of a high-level description. This could be a narrative, and we came up with a formal way of specifying this high-level behavior. So this is a program. We have a programming language that we're using, and importantly, this programming language is not at the level of genes or promoters or things of that nature, is rather at a higher level. Just like when you program a computer, you're not going to program that at the level of the basic logic gates. Rather, you try to express things at a higher level, whether it be subroutines and much more elaborate specifications that then you can wire together in different ways. So that's where we see the future of synthetic biology is at a high level of specification, and the idea is that the compiler brings it all the way down to genetic network, looks at DNA parts, and then sends that off to a robot, which then figures out how to put this together, and ultimately you can put this inside the cell. So we are working on full automation of this, and I'll show you. And then obviously we want to understand how to iterate over the designs over here. So let me give you a quick example. So here's an example. And this is a high-level specification. So some of you, I don't know how many people here have experience with Lisp or Scheme. So that's why we're using all these confusing parentheses and so on. But if you like something like this is a simpler, maybe to you, notation. So if the input AHL is high, produce cyan, else produce yellow. So that's what we mean by high-level specification. And I'll show you more and more complex specifications later on. And so the idea is the compiler takes this and generates a data flow where we have boxes over here representing HL as an input, goes to cyan, the decision to make cyan, that gets flows into another box which inverts it, and the decision to make yellow. Okay, so we can take this high-level representation, create a data flow, and then the compiler automatically makes an abstract gene network that implements this data flow. And notice that some of the things are actually specified, but some are not specified yet. And then we have a tool called Matchmaker as part of our collaboration with Jake Beal and Doug Densmore that takes this abstract genetic regulatory network and actually decides on an actual sequence based on the parts that are available and creates a sequence when, for example, this repressor here is actually chosen at that particular time to make sure we satisfy the high-level specification. And then that gets put into a set of instructions for assembly that is done by a robot. So with respect to this flow, we have from here to here, we have this fully automated, and also we have some portion of this working as well. So I would expect within about 3 months or so to have end-to-end compilation from a high-level specification to something that gets fully automated and built by the robot. And this is in fact a robot that we have now in our lab. that does a lot of this. So our BioCompiler can do many things. So it can do combinatorial logic, so you can specify any kind of logic function, and it can create a gene network, and then we can simulate that gene network. We can also have state in our compiler. So, for example, if we want to have this, the system or the cells store information, we can provide that. And actually, it provides spatial computing as well. And this is how I got teamed up again with Jake Beal, and they came up with a high-level specification. And I show you the pattern formation over here, and they came up with a high-level program that allows you to describe this spatial organization over here, but not at the level of the individual cells, but rather at the level of the overall cell community. Okay, so that's a really cool thing about this. You can specify it at a high level, and then this gets compiled automatically into something that gets implemented inside each individual cell, and then you can go ahead and simulate that. And then ultimately, obviously, put that inside the cells for actual implementation. What was interesting is that this circuit right here that was automatically compiled from this code is actually essentially identical to what we designed in the lab by hand. So that was quite interesting. So let me switch gears now. So that I showed you about the high-level compiler. One thing that we're actually working on implementing now is the notion of transient behavior as well, and that has to do with the next example. But what I want to show you now are a couple of examples where we're now not just trying to turn GFP on and off, but trying to create applications where hopefully there might be some kind of a usage for them, perhaps clinical. So in this case, the first case is where we have cancer therapy, and probably the biggest issue right now with cancer therapy is the notion of specificity. Okay, so you can have therapeutics that are in fact able to kill all the tumor cells, but the issue is that at the same time there'd be a high level of collateral damage, and they will destroy the healthy cells as well. And so, for example, you can have a therapeutic agent that might recognize something on the cell surface of a cancer cell and deliver the toxin to the cell and kill that cell. And what we argue is that there's simply not enough information here that's being used. For example, just looking at a single cell surface receptor, we believe in general is not sufficient. And that's why our approach, we are taking this therapeutic agent, having that go inside the cell where all the information that you need is in fact available to make a highly precise and specific decision about whether the cell is cancerous or not. And I'll show you how we do that. And so the idea is to use RNAi-based logic circuits that can take multiple inputs, use these multiple inputs to make this highly precise decision. And let me also note that even if this is not the cure for cancer, there are many kinds of non-clinical applications for this. For example, for development, for understanding of various kinds of processes that are taking place over time. So we focused on Hela cells, and we built a cell classifier, and this is work in conjunction with Kobe Benenson, who's now at ETH in Basel. And so we focus on Hela cells, and again, the idea is that we will have a therapeutic agent that goes into all of these cells and decides whether each individual cell is a Hela cell or not, and kill it if the answer is yes. So that's high-level specification, detect and kill Hela cells. And here's the program. So it looks a little bit ugly, I guess, with all these notations, but it's actually rather simple. These are ands, these are nots, and all this is saying is that if these inputs are high, and now these inputs are microRNA that are present inside the cells. Okay, so we look at the endogenous microRNA levels, and we decide what is a pattern of expression of microRNA that distinguishes Hela cells from all other cell types. And we believe this one here is in fact the one for Hela cells, and we also believe that you can generate that for any cell that exists. Okay, it might be much longer profiles, but we do believe that something like this exists. So here these microRNAs have to be high, these have to be low, and that's a Hela cell. Okay, so how do we go about creating this classifier? So we have to have parts. So for example, here's a part that basically is on when the microRNA level is low. And this is very simple. The microRNA simply represses the output. Okay, so that's what we have to do. And now we can combine this basic sensor with multiple sensors. And so now we have an AND gate of these multiple sensors, and we just combine that by fusing it alongside at the end of our output over here. Okay, so the output is high only when all of these inputs are low. Okay, so that's an and of knots, if you will. Okay, now we also have a high sensor, meaning that the sensor is high when the input is high. And so what we do is simply do a not of this input right here that represses these intermediates. These are, it's a transcriptional activator and a transcriptional repressor that is activated by this activator, which represses the output. And see here, what you're seeing is the gene network representation with the RNAi interactions as well. So we can take a sensor like this, a high sensor, and we can also combine that using an AND gate. And the way we do that is we simply tie together the two sensors to have the same output. And we kind of got maybe lucky in a sense that in this particular case, for the sensors, for the way these sensors are actually engineered, we can combine these sensors and have them still have the same transcription factors, but the difference is that they have different regions here that sense different microRNAs. Okay, so the output here is high only when these two inputs are high as well, and in all other cases the output is low. Okay, despite the fact, and I can talk about this later, despite the fact there's in fact crosstalk over here, which actually does not hurt the operation of this particular AND gate. So we can take this AND gate that you're seeing here, and then we combine that to form a longer AND gate with the microRNAs that are supposed to be low. So this is the full circuit that we implemented. Okay, we tested this circuit, and here are the 32 different input combinations that we have. And you see that the output is high only when we have the appropriate microRNA expression pattern, okay? Or we provide the microRNA from the outside. And you see here we actually had some basal expression that we noticed in 3 different cases. And what we did was we engineered a slight modification I won't go into, where we engineered a feed-forward motif with RNAi, and then in fact reduced the levels that we're seeing here. We then tested this circuit on 7 different cell lines. So here are the 7 different cell lines that we tested. These are the Hela cells, where we see the positive, the high response. And these are the responses we see in other cell lines. Notice that we actually did not engineer the circuit against these cell lines. These are cancer cell lines, other cancer cell lines that we use because they're much easier to work with than primary cells. But in fact, we still see a high level of specificity. Okay, and the case here where we have the HEC293 cells, we figured out that the reason for this was that the promoter that we're using was actually pretty active in HEC cells. But if you see in the downregulation, in terms of relative knockdown, it's still really good. Since then, we've actually optimized this circuit further. This is done with multiple plasmids for transfection. We actually created a single plasmid, we transfected the single plasmid, and we got 25-fold difference between the Hela cells and the HEC cells. So we're quite happy with the experimental results in terms of specificity. We tested this as well for killing efficiency, and here you see these are the Hela killing, and this is the HEC293 killing, and we see that it kills almost as good as constitutive killing, and then here for the HEC cells, we see that it really doesn't harm them too much, even with this multi-plasmid. And now we're testing that out with a single plasmid. So there's selective killing even against the worst case cells that I just showed you. So let me switch gears to the last example. And in this particular case, we're looking at artificial tissue homeostasis. And so we're trying to engineer a system that might be relevant for diabetes. This is one of the holy grails. So we're right now in the computational design, and I'll show you some experimental results from implementation of modules. But what we're trying to face here is the notion that in diabetes type 1, you have the autoimmune system that slowly kills the insulin-producing pancreatic beta cells. So we'd like to be able to create a system that maintains the beta cell population despite the fact that we have this autoimmune attacks. Okay, so what we'd like to see is a situation where, for example, we engineer stem cells, we transplant them, they maintain their own population, and then when needed, they differentiate into beta cells or beta-like cells, and then when there's enough of those beta cells, we make sure that the stem cell population does not produce more beta cells. So it produces the beta cells as needed. Okay, and so we obviously don't have this entire system working at this as a huge system. 22 components. We're trying to understand how to create this both for the potential therapeutic reasons, as well as trying to understand what are the issues in terms of the system level design of something of this level of complexity. Right now, I think that the biggest thing that's been published has been about 6 parts or 6 components or so, and we're moving higher than that. But the question is, what kind of challenges arise when you go to something of this level of complexity? So experimentally, I have one slide showing some results. So we have been able to create a module that differentiates embryonic stem cells with a program to cells that have beta-like properties. Okay, they exhibit many properties that are associated with beta cells. In fact, they're able to produce insulin. Okay, so we've had some success there. We have cell-cell communication working in mammalian cells as well. And this is actually by integrating AHL and having enzymes that produce AHL and receptors that respond to AHL. And we've also been able to implement things like a toggle switch. So, for example, so if the cells decide that they need to become beta cells, hopefully they will actually remember that decision. Okay, so experimentally, we already have many of the components that we need to create that high-level system. This is what we'd like to do with all those components. Okay, so this is a big program. This program is supposed to implement the algorithm that I just mentioned to you, and what we're working on now is getting the BioCompiler to be able to automatically compile this program to a gene network that implements this. What I'll show you now, though, is how we did this by hand with the aid, and then I'll show you the aid that we're using with the computational modeling and trying to understand what are the properties of the system. So we create a system architecture like this at this moment by hand, and the idea is that the stem cells maintain their own population with cell-cell communication, and when the stem cell population is high and the beta cell population is not high, here we have an AND gate, then a cell, this is an individual cell, is supposed to make the decision to differentiate. Okay, so that's the idea over here. And so we said, okay, this looks like a reasonable design. We modeled the system, and we noticed that computationally, sometimes the system, in fact, maintains a population of beta cells. Also, that's the dashed red line. Also of stem cells, but it's okay if the stem cell population oscillates it a little bit. But that's under some set of rate constants. But if the rate constants change, then the system might actually oscillate. So this is not an ideal design. So this is version 1. So version 2, we try to remove these oscillations. And so what we did is we noticed that there's a long feedback in terms of there's a long delay between the decision and the final differentiation of the beta cells to the time that they become beta cells and prevent further commitment by stem cells to differentiate. So now we have a fast toggle switch. I actually showed you experimentally that this is working in the lab. So now we have a fast toggle switch. Cells commit to differentiate, and a cell makes a decision about whether to commit to differentiation based on the uncommitted population, these are stem cells, and a committed population. These could be either stem cells that are committed to differentiation, or actual beta cells. Okay, so this is the design number 2, and the idea is to have fast feedback that basically decouples the differentiation process. Okay, so we did a computational model of this. We saw the computational system dynamics look nice. The system is able to create a steady level of committed cells. If we do all kinds of stability analysis, we have an attractor state. So everything looks good with respect to this design with ordinary differential equations. Then we integrated noise into this, and it's a good thing we didn't actually put this into patients just yet. And so here what you're seeing is that we have large fluctuations due to the noise in the system. So this system does not work just yet. Okay, so we came up, let me just show you what this looks like. So this is, for example, a simulation that we would do, and you see the uncommitted population going up, committed population going up, but then the system is not able to maintain a steady level of the committed population. So it never quite crashes, but it's not able to have a nice steady state. So here's system architecture number 3. And in this case now, we're trying to understand how to create a more robust system. And so one of the things that we often have to deal with in biology or in synthetic biology, and certainly in biology as well, is the notion of noise. right? So how do we handle noise in the system? How can we take these noisy biological components and create reliable systems out of it? For example, reliable digital systems. And what we said here is, rather than trying to fight noise, we're actually going to use noise to make a more robust system. And specifically, the way we did this is by integrating an oscillator. And so oscillators have actually been built in synthetic biology. And what happens is they rather quickly go out of phase. And so that's been an issue, is trying to create oscillators that are actually in phase. And there's been some nice work by Jeff Hasty, for example, showing that you can actually do that with cell-cell communication. But we said, well, actually, that's great. We want these kind of quote-unquote bad oscillators, because what happens is, as soon as they go out of phase, we use that as part of our AND gate, such that only the oscillators that are high, only cells that have an oscillator that's high, are in fact allowed to differentiate. Okay? So we have a 3-input AND gate with that controls the switch over here. So we're exploiting the biological noise to break the symmetry in our system. So we in fact simulated this, and with a noisy simulation, in fact, it looks like the system is able to maintain homeostasis. Okay? We did computational modeling to understand what rate constants influence this, and you can figure out what rate constants. Something that we thought about is, well, what we do in synthetic biology is we often create modules first. We spend a lot of time in creating the modules, optimizing them until they actually work. So if we were to create an oscillator, if we were to create a quorum sensing like an AND gate, a toggle switch like here, it takes a lot of effort to create each one of these and have it functional. How do we know that it's actually going to give us the appropriate behavior when we integrate all of these? And so that's why we think that looking at the individual modules and specifically at their phenotypes, rather than the rate constants of the individual elements, is the right way to go. Okay? And so we did, in fact, computational studies to try to understand which phenotypes and what values can actually help us get the best signal to noise. That's a measurement of the performance of the system. So, for example, the duration high, that's for the oscillator. So we want to optimize particular durations for the oscillator as opposed to really worry about the rate constants first. So this is, we believe, the appropriate level of abstraction. And we, in fact, did an analysis to try to understand the importance of the rate constant in being able to predict the overall system signal to noise or performance versus the module phenotypes. And it does, in fact, it is the case that the module phenotypes are much better predictors of overall system behavior. And so this is a system, and this is a simulation of the system. It's able to maintain homeostasis. And see this is, okay, so I'm actually just about done. This is the overall system design. So this is, as you can imagine, quite a huge undertaking. But we believe it's the right time. So I showed you very briefly cell-cell communication. This is for the quorum sensing. We have a working toggle switch. Cell differentiation to cells that appear to have beta cell properties is also, looks like that's working. Oscillators have been implemented in mammalian cells. For example, by Fusenegger at ETH, we're working on an oscillator. Pam Silver implemented one. These are all in mammalian cells. We are actually working on multiple cell-cell communication mechanisms. I showed you a 6-input AND gate. So really now is the time in synthetic biology to think about the system integration issues. How do you take all of these modules and actually integrate them? So we implemented a variety of basic modules. We're now pursuing applications that are uniquely enabled by synthetic biology and exploring the system design principles. Let me show you actually a different perspective about this in terms of the outlook. So today, we use models to guide our designs. We don't have a perfect ability to predict what happens. What we do have is the ability to correlate various modifications, for example, in rate constants, with the system response. We have part characterization, some working small modules, and so on. Well, very soon we're going to have is end-to-end ability to specify behaviors at a high level and get that automatically compiled and assembled by a robot to a circuit, and then understand the composition rules. What we'd like to be able to achieve in the future is certainly a better understanding of how the circuit is integrated with the rest of the cellular context. And so this is a huge, huge challenge, and certainly systems biology has to come in, has to play a very important role in this particular goal, and then ultimately be able to do predictive design. So I will stop there. Many people in my lab to thank. I've also mentioned work that we have in collaboration with Kobe Benenson, work with Jake Beal, and also Doug Densmore as well. I'll stop there. Thank you.